Good morning. I think, I'm 1 minute early. I guess we could kind of get started because most of the announcements are housekeeping things like, what to do with your phone. So anyway, I'm Jane McPherson. I'm a member of the HAS Science, Medicine, and Technology Committee, and we're really happy that you could be at today's session about immunotherapy. I'll get to introducing our speaker in a minute, but I want to remind everybody, turn off your cell phones, turn them down, do whatever you have to do. And if you have a very deep purse, make sure your cell phone's on top in case it goes off despite your efforts. And then welcome to the crowd joining us from Zoom. And Gloria Goodwin for being our course tech over there. So that's a very scary job, I think, but she's doing a really good job. And then one other announcement today's Wednesday. Today, there's a sort of a bon voyage to Ian and a welcome Amy in her new role party this afternoon from 2:30 to 4, I think, yes, here in the HASP office. As many of you know, Ian is moving on to a wonderful opportunity for him in the Grand Rapids area, and Amy is being promoted. So we'd like to, wish them our best in both capacities. So please come if you can. And today, our speaker is McLane Watson, who goes by Mac, or at least you do in your emails, right? Okay, all right. And he is from Boulder, Colorado initially, but I was eavesdropping. It came to Michigan to go to Blue Lake Music Camp, right? Because he plays cello. And he spotted Holland and fell in love with Hope. And so he did his undergraduate here. So that's how he became a Michigander. And now he's married, and his wife is also from the area. And they loved it so much, his parents moved here. So he's really got a family affair going here today. He did get his undergraduate degree in biology from Hope, and then he went to University of Pittsburgh, where he got his PhD in immunology, and now he's working at the Van Andel Institute doing a postdoc fellowship, and has been there since 2022. So we welcome you today. As far as questions, Max said it's fine. Today's is more of an intro to immunology so that you have the sort of setup and how it would be used to treat cancer. So if there's questions, feel free to ask during the presentation. I know a lot of times we hold till the end, and we still will take questions at the end. So if you don't want to interrupt, that's fine. But sometimes you might feel like you're missing something, and I think that'd be reasonable to ask. Yeah,
absolutely.
Thank you.
All right. Hello, everyone. Thank you so much for having me. I'm excited to teach you all about immunology and cancer. That was a very wonderful introduction. I wanted to give a little bit more of my background so you guys can get to know me just a little bit more. Like I said, I'm from Colorado, technically Louisville, but it's like 10 minutes outside of Boulder. So I usually just say Boulder for people. I majored in biology here at Hope. I also minored in music, playing the cello. So after I graduated, I got a job as a lab technician at the Van Andel Institute, working on prostate cancer research. And it was really in that lab that I thought the next kind of wave of treatment of cancer is going to be with the immune system. So people have been trying really hard, kind of avoiding the immune system in classic cancer biology for a really long time. And then kind of around 2011 was when the first immunotherapy was approved for cancer, and it really changed the game. And we'll kind of go into that more in the lecture. But so I sought out a PhD in cancer immunology, and I ended up at the University of Pittsburgh. And I researched cancer immunology, these specific cells called Treg cells, which we can get into a little bit later, but how they interact with the tumor and how you can kind of enhance immunotherapy by altering their function. And so in 2021, I defended my thesis, and then I looked for a postdoc opportunity, and I didn't think I was going to come back to the Van Andel Institute, because when I left, they really didn't have any immunology going on. But they brought in this guy named Russell Jones, and he is an immunometabolism person. And so I got super excited that I could get an opportunity to come back. So I asked if I could join him in his lab, and he said, of course. And so now I've been doing a postdoc for about 2 years. And during this time, I've also received a Damon Runyon Cancer Foundation Fellowship, which kind of recognizes up and coming scientists and supports kind of high risk, high reward research. So that's where I'm at now. And so I feel like I'm qualified to be able to speak to you all about cancer immunotherapy, as that's been my background. And yeah, as Jane said, I can take questions as we go along. I'm trying to give kind of a broad overview. My goal here today is to, and actually let me go to this outline. First, I kind of want to define immunotherapy and cancer, and for the majority of today, really talk about the immune system and what are the players of the immune system, so that those of you joining next week, we can also talk about how those actually interact with the therapies and how the therapies for cancer are playing with these different components. So today is kind of a background, but definitely still sprinkled in with lots of cancer immunotherapy. So we'll go over what the immune system is, kind of innate versus adaptive immunity, these cells called B cells and antibodies, T cells, some 3 E's of immunoediting, and then some barriers to cancer immunotherapy. So cancer immunotherapy, probably heard of this term, but many of us maybe don't know exactly what these things mean. So what do I mean by immunotherapy? Immuno obviously is involving the immune system, and therapy is a treatment intended to relieve or heal a disorder. So immunotherapy is harnessing the immune system to treat a disorder. And in this case, the disorder is cancer. But immunotherapy doesn't have to be for cancer specifically. It can be used to treat a wide variety of diseases, especially autoimmune diseases. There's many therapies out there that are immunotherapies that are altering the immune system or harnessing the immune system to treat those as well. So we know what the immune system and immunotherapy is, but a lot of us have heard of cancer and know of people that have experienced cancer. But what does this actually mean in terms of the biology and cells? So how do we define cancer? Cancer occurs when cells divide uncontrollably, and this uncontrolled cell growth can lead to tumor formation, right? So at the very basis of what cancer is, it's just uncontrolled cell growth. However, uncontrolled cell growth alone doesn't necessarily make cancer super deadly. So what is it about cancer that makes it deadly? And it's really the metastasis part. So I don't know if you've all heard of the word metastasis, but uncontrolled cell growth is bad on its own. And here, let me use this pointer. But if it's contained, it can kind of be dealt with. So if, you know, I guess the easiest representation of this would be if you have kind of a funky looking mole on your skin that could be potentially melanoma. Usually the doctors will say, well, does it have a defined border? If it has a defined border, that's a good thing, because that means it's not invading into other tissues. And that's really where cancer becomes a problem, is when it starts to invade. And so essentially metastasis is the fancy science term for invasion. So in metastasis, you can have a primary tumor site. So let's say you have a lung tumor, but then it starts to invade other parts of the lung. It can get into the bloodstream here and then travel to distant sites, and then come out of the bloodstream and then like into the bone, for example, or into the brain, and start to grow and form a tumor there. So when that happens, that's when it's the problem, because all of a sudden you have tumors that started off in one place and then have disseminated all over the body. And then when those out of control cell growth starts growing everywhere, then that can be a problem for organ function. So it can shut down your lungs, it can shut down your brain, and then that's what would lead to death. So let's see here. So that's right. When people talk about they have a lung tumor, usually the lung tumor part is referring to the primary tumor. And that tumor can go other places and end up in your brain or skin or bone. But usually people talk about cancer as the primary site. So if it started in the lung or it started in the pancreas and then it went to the bone, now all of a sudden you don't have bone cancer, you just have lung cancer or that has metastasized. All right. So cancer is also kind of this multi-stage event that it doesn't just all of a sudden happen where you have these cells that go rogue 1 minute and start producing a tumor. So in this kind of cancer progression, we have normal cells here. And then there's usually something that mutates or silences genes that regulate cell division. So because cancer is an uncontrolled cell growth, this makes sense, right? If you have mutations in genes that promote cell growth, that could drive the cells to divide. If you have mutations in genes that stop the cell from dividing, if you take away that barrier, those cells can then uncontrollably divide. But again, kind of, I like to envision this kind of most easily as skin cancer is kind of the easiest to visualize. So when you have UV damage from like sunburn or just prolonged exposure to UV, that can mutate things, but that in itself is not going to produce cancer. So you might end up having that mutation as the initial initiation stage. However, then there's some other signal or event that occurs that causes that mutated cell to divide. So initiation, you have some mutation in a gene, and then promotion, you push that cell to actually divide. And then once it starts going, those things that regulate that cell division, because they're mutated and no longer in place, then the cancer, then the cell can kind of go uncontrollably. And then as that cell keeps dividing, it's not a normal cell. So the things that normally keep cells in check when they replicate their DNA to produce daughter cells, those are kind of all out the window. So you get more mutations, which can lead to more uncontrolled cell division, and then eventually a malignant tumor where it can start to invade other tissues. So I kind of have a question for all of you. Aging is actually one of the #1 risk factors for developing cancer. And based off of this paradigm, why would you think that is? Yeah, essentially opportunity, right? Because there's many years to be able to accumulate mutations, right? Either again through skin damage or we're exposed to different carcinogens or whatever it is, right? And then that time also allows for whatever that promotion stage will push that cell to divide. And so as you age, you accumulate these mutations, and those by themselves are not necessarily bad until something pushes the cell to divide, and then it can get out of hand. Awesome. Okay, so just to summarize cancer. So cancer arises when cells divide uncontrollably. And then metastasis is when the primary tumor cells invade and then grow in other tissues. And this is really what makes cancer deadly. And then cancer arises in stages: initiation, promotion, and progression. Okay. So we've also in the recent years have heard a lot about the immune system. So I wanted to throw it out to all of you. What do you think when you hear the word immune system? And you can kind of shout it out if you have ideas. Defense, yeah. Something we don't know very much about. Yeah, that's good. Yeah, very good. Any other thoughts? I always think of like, whenever I tell people I work on the immune system, like, oh, I have a great immune system, or I know this friend that has a horrible immune system. A lot of times people will talk about allergies too. The term white blood cells, I think we've all heard white blood cells and red blood cells. But I feel like these are terms that are kind of ubiquitous in our society, but like we maybe don't know specifics about what we're talking about. Maybe you've heard of autoimmune diseases, diseases that attack your immune system, attacks your body. Vaccines too, as we've heard a lot about. And of course, like colds, viruses, bacteria. So in a broad sense, the immune system is this complex network of cells, tissues, and organs, and the substances that all those make, and that helps the body fight infections and diseases. So really, it is the body's defense system. It is the body's ability to maintain homeostasis. And this is my kind of own definition, is that the immune system is made up of many different cell types, all playing an important role in keeping us healthy and free from things that infect us and any potential cancer that arises. And we'll talk more about that. So the immune system, as you can kind of see by this diagram, is split up into two main parts. We have the innate immune system, and then we have the adaptive immune system. And you can see that each one of these little circles is a different cell type. So really, this is a network of cells all working together to try and keep you healthy, try and keep your body at homeostasis and prevent infection, and also promote wound healing too. So they're not just fighting infection. So they do a lot of important things. So first, I'm going to talk about what the innate immune system is. So I said the immune system is kind of broken up in these two big arms. We have the innate and the adaptive immune system. So the innate immune system is made up of cells, these cells, and we'll go over them, but also physical barriers. So you can think of your skin is actually part of your innate immune system. As long as your skin is intact, you know, if you get bacteria or encounter other things, also, you know, mucus in your airways traps a lot of dust and germs and things like that, that will just prevent things from getting into your body in the first place. So that's actually a really important aspect of your innate immune system that I think a lot of people skip over. But if they make it past those kind of physical barriers, we have cells to be able to take care of that. And some of those cells you may have heard of, like macrophages or neutrophils, natural killer cells, mast cells. These are all part of your innate response. And so, like I said, when they get past that first barrier, these are the first responders. So these cells sit in all your tissues, in your skin, in your mucosa, so your eyes and nose and mouth, anywhere that could potentially be infected, even, you know, the back of your throat. You think of when you get a cold and you have that sore throat. So these will be the first responders to infection because one, that they're in the tissue, and two, they're very quick. We'll talk about the kind of differences between innate and adaptive in terms of timeline, but innate is very quick because, again, they're there and then they can send out signals to recruit other cells to come very quickly. So these cells are kind of constantly patrolling the environment and looking for any danger. And so the innate immune cells have several functions, kind of three big functions. One of them is to eat foreign invaders. So a perfect example of this is a macrophage. We have macrophages again in all sorts of tissues, even in our lungs. And let's say they encounter a bacteria, they will literally latch onto that bacteria and then engulf it and eat it and destroy it. So that's of course a very good thing. We don't want bacteria just running amok in our system that aren't supposed to be there. And so that makes sense. They're very direct killing and just eating and destroying invaders. But the other thing that they do is they send out signals to recruit other immune cells to the site of infection or whatever they encounter. So they might eat something and then they realize that what they've eaten is actually not supposed to be there. And so they will send out these chemical messengers that can get into the bloodstream that then actually bring in a lot of these other cells, especially neutrophils, maybe more macrophages, that will kind of swarm the area and take care of any other bacteria that might be hanging around. And then really importantly, they also take samples of whatever the invader is. And I'm using invader as a term for this because this can be viruses, this can be bacteria, and this can even be applied to cancer cells, because cancer can be, even though it's self technically, it becomes foreign in terms of the immune system because those mutations make it look different than self. So they can take samples of whatever the invader is, and then they can come back and present it to the adaptive immune system. And we'll talk about the adaptive immune system in a second, but this is a really important thing to be able to control infections because so sometimes the innate immune system can completely take care of the infection on its own, and you don't need the adaptive immune response. But a lot of times the adaptive immune response gets involved because the innate immune system can only do so much, and then it needs the help of these kind of specialized cells. So here's a good example. The innate immune system is activated by these kind of danger signals. So in this scenario, let's say you're preparing dinner and you accidentally cut yourself, which can happen all the time. But if you're, cutting raw meat and there's some bacteria on the knife, all of a sudden you have this open wound, it's gotten past the skin barrier, and you have bacteria enter your skin. And so those innate immune cells that are in the skin will immediately respond and notice that these bacteria are foreign. And we'll talk about that on the next slide of how they recognize that they're foreign. But they'll eat these bacteria as much as they can, but then they'll also send out signals, and these signals are also known as cytokines. So they send out these cytokines, which are these chemical messengers that can then enter the bloodstream. It can also diffuse throughout the tissue and then recruit other immune cells to this site. And so we've all probably experienced this where you get some sort of injury and then it feels, we use the term inflamed, right? Where it gets very red, and it also seems to swell and all those sorts of things. And so that swelling, that redness is all caused by this cascade where these cells are producing these cytokines, which recruit other things, and also open up the blood vessels to allow more immune cells to come to that area. And so that's why you get this like redness and swelling, is because literally kind of flooding the area with immune cells. So the way that these innate cells respond is termed non-specific. So they'll see foreign substances like a bacteria, like a virus, but they're not specific for any particular bacteria or any particular virus, but instead they recognize patterns that are common to invaders. So I like to think about this in terms of cars. might have several car references here. I like cars, so the innate immune system recognizes patterns. So here's 3 different cars, and we can all appreciate that even though these are very different, they are still cars. I mean, one could argue that maybe not this one, but we can see that they have, 4 wheels. They have a windshield, some mirrors, an engine, lights. If we can see them moving, they move forward, they move backwards. You can turn a steering wheel, right? So those are all patterns that we can recognize that these are cars. And unlike the innate immune system, we can tell that these cars are different. Like, this is definitely a supercar and this is not. So the innate immune system would basically just see that all of these are cars because it can recognize the patterns of the 4 wheels. It can recognize the windshield, things like that, but it just sees it as a car, which is good enough for the innate immune system. It just sees that it's an invader. So like bacteria, even though there's a wide range of bacteria that can infect us, or viruses, there's a wide range of viruses that can infect us. The innate immune system has these receptors that kind of just recognizes commonalities of like, oh yeah, this is an invading bacteria, or oh yeah, this is an invading virus. Not that this is COVID-19 or RSV or E. coli or whatever specific. Okay, so just to summarize, the innate immune system, they're the first responders. They are tissue resident, as we say, and then they recognize patterns, so they're non-specific. They don't go after particular viruses and antigens, but just they are, as a whole, can recognize invaders. And they're very good at eating invaders and recruiting other immune cells. And a lot of times, this might be good enough to wipe out any potential infection that's coming in. And you probably wouldn't even notice that you were that sick if you had it with just the innate immune system was able to take care of things. So this is where we get to the adaptive immune system. So what is the adaptive immune system? I liked this cartoon. It definitely anthropomorphizes the cells, but I think it gives a good kind of representation of what these cells all do. So the adaptive immune system takes over when the innate immune system can't control and destroy the invader. And what's different about the adaptive immune system is that these are specific for whatever virus or bacteria that you're actually infected with. So if we go back to the car example, they would be able to tell that this is a McLaren versus a Toyota Prius. Like they're specific for those types of cars, essentially. So we have T cells or B cells that can only recognize COVID, RSV, flu, strep throat. So they're very specific, but this is a really good thing because that means they're very good at finding and killing those viruses and bacteria. But the downside is that the specific recognition takes more time. So these T cells, I can't remember what my next slide is. We'll maybe go into this a little bit more, but specific recognition takes more time. So you have to kind of find the right immune cell that recognizes the thing that has entered your body. And then that immune cell has to multiply and grow itself and then move from where it was to the site of infection. And so usually this is on the like order of 7 to 10 days that your immune system, the adaptive immune system, will start to kick in. So your innate immune system can kick in within hours and like the first day, right? But then adaptive immunity takes a little bit more time. And probably the most important part of the adaptive immune system is that it can remember invaders encountered. So this is the concept of immunity. Just like you've all had chicken pox when you were little and you don't get it again, that's because your body remembers that you had chicken pox and has this kind of system and defense already set up so that if you ever encountered chicken pox again, it could quickly wipe it out. Okay, so what makes up the adaptive immune system? We have kind of broadly these three things. So we have T cells, and this is actually, I mean, this is colorized, so it's not the actual color, but this is a scanning electron image of a T cell. So you can see, I mean, it's basically a sphere, but then it has like all these crazy little indentations. And then we have B cells, which we'll talk about next. These are the antibody producers of the body. And then, of course, we have the antibodies that the B cells produce. So B cells, as I just mentioned, are the antibody producers of the body. I'll go into more detail about what antibodies are later. But first, so most people, and including me, before I actually looked up this lecture, when I hear B cells, I've been asked like, oh, where does the B come from? And I would have said it came from bone marrow because these cells develop in the bone marrow. However, they don't actually come from bone marrow. They come from this bursa of Fabricius, which is actually in birds. And this part of the chicken was first described by Fabricius in 1600s, but it wasn't actually until 1950s and 60s that researchers at the Ohio State University, it was actually two separate researchers. One, and I can't remember why, was bursectomizing, so taking out the bursa Fabricius from these chickens and then doing some sort of experiment. Another researcher was like, oh, I want to produce antibodies against salmonella. And so I'm going to go and immunize these chickens and then, get my antibodies back. And he went to immunize, he had these bursectomized chickens left over. And so he took those, he injected them with salmonella, and they all ended up dying. And he's like, what the heck is going on? And the ones that maybe did live, there were no antibodies. And so kind of fortuitously, they figured out that whatever cells were coming from this region in the chicken were producing the antibodies. And so, yeah, like I mentioned, in humans, we do not have this. We have bone marrow. So that's where our B cells come from. But I thought it was just interesting that B cell does not stand for bone marrow. So B cells, again, specialized cells, and they're, to me, if you ask a B cell biologist, they probably hate this, but to me, they just produce antibodies. That's really the important thing. And so what is an antibody? Antibodies are incredible little proteins that float around in your bloodstream, and they can quickly detect and attach to foreign invaders. And so the recognition of these antibodies to kind of the foreign invaders is like a lock and key. So here you can see there's a kind of a bunch of different kind of puzzle pieces, if you will, and only one is able to fit into this kind of recognition site. And I depict this antibody this way. It kind of looks like a Y because in real life, that is actually what it looks like here. If I go back, here is the protein structure of what it looks like. So these kind of ends over here are the same thing that's recognizing that puzzle piece. And so what's great about antibodies is they're able to specifically recognize a part of a foreign invader. So going back to the kind of car analogy, not only can they recognize that it's a McLaren, but they would actually recognize that it's a 2013 McLaren and I don't know, made from this factory. Like they're that specific that they can see that kind of detail. So really it is more like a lock and key, where a key is really specific for only one lock. That's what antibodies are. And so you can have antibodies that recognize multiple portions of the same invader. So again, that's kind of how specific they are. So like for COVID-19, you could have one that recognizes the spike protein, for example, or you could have one that recognizes another part of the capsid, but they all recognize COVID-19. It's just specific bits of it. So whenever I was learning about antibodies, I was like, okay, that's cool that they combine stuff, but what does that actually do to help destroy invaders? So really they have three main functions. They can neutralize invaders, and we'll go into detail on each of these. They can activate other immune cells, and then they can activate proteins in the blood. And I actually really like this image. This is another scanning electron microscope image of, if you see all these kind of white fuzzy looking things, those are antibodies coding some sort of invader. So you can really see that they're like literally creating this coding around whatever it is they're specific for. So one of the big functions of antibodies is to neutralize invaders. And I like to use COVID-19 because we have kind of context for that. So here we have the COVID-19 virus, and it has all these spike proteins around the outside. And how COVID works and how many other viruses work is that these spike proteins usually bind with some receptor on our cells, and then that allows the virus entry into that cell, and where it can divide, produce more of itself, and then kind of spread to other cells from there. So this binding is kind of key for its entry into the cell. And so antibodies against COVID-19 can literally block this interaction and bind up that spike protein so that it can no longer interact with this receptor and then gain entry into the cell. So if you can't gain entry, then you can't make more virus. So another thing antibodies do is that they activate other immune system cells. So I'll actually pull this up because here's that picture again. What's really cool is that the antibodies will literally coat the invader. And here's kind of a cartoon depiction where they kind of, can coat the outside or link up a bunch of other ones together. But this bit that's sticking out at the end, the bit that's not binding to this invader itself, actually can interact with immune cells and receptors on those immune cells. And when this interaction occurs, those immune cells see that as like a eat me signal. So they'll see that and then they'll kind of engulf and literally eat it. And so while immune cells can eat other invaders by themselves, this makes it much more efficient and easier to know that, oh, this is definitely an invader. It's covered in antibodies. If you can think about it, it makes it more delicious for them to eat. I don't know. Okay, and then this was actually something that I had no idea about before I learned some things about immunology, is that we have these proteins in our blood that are constantly circulating, and they're called complement. I don't know why they're called complement, but these proteins, what happens is those antibodies can bind. Let's say this is a bacteria cell. Those antibodies can bind to whatever they're specific to, again, coding that. And these proteins that are in your blood, called complement, can also recognize this little non-specific bit that's sticking out from the antibody. And when these bind together, that starts this cascade of signaling, which is termed complement activation. And there's a lot of steps to it, but eventually what happens is that it makes this membrane attack complex, or MAC, and this inserts itself in the membrane and essentially just creates a giant hole. And I mean, you can think about a balloon. If you create a hole in a balloon, it's going to pop. Or, you know, these things are surrounded by kind of water and liquid, that if you punch a hole, water is going to come rushing in, other things are going to come rushing out. And so you essentially can neutralize and kill the bacteria in that sort of way. So I thought that this was really interesting, that we just have these proteins hanging around. Okay, so to kind of summarize, let's see. So to summarize antibodies, and they have three functions. They can block invaders from coming in. So literally just hindering physically the motion and activation of those. They can help other immune cells eat them and signal to them to eat them. And then they can activate proteins in our blood that punch holes in invaders. So big question is, why are B cells and antibodies so important? So of course, B cells and antibodies are important because they help destroy invaders. But after the invader is gone, B cells continue to work. They don't just stop working. They continue to work by becoming memory cells. And so these memory cells produce very potent antibodies that will circulate in your blood and act as a defense mechanism for if you get reinfected with the same thing. So again, these are very specific. So, you know, we get colds probably every season. And even though that cold is like a common rhinovirus, it's still a rhinovirus every single time. It's just slightly different than the last time. And so you would think our bodies would create memory against that, and they do, but it's very specific for that particular rhinovirus. So usually if you get a cold in a season, you might not get a cold in that season again, even though you encounter it, but you have this memory against that specific thing. So I wanted to kind of go over this schematic. When we were talking about innate immune system, this is, again, like response on the order of like hours to a day or two that they will respond to some sort of virus. And here, this is just showing on the y-axis, this would be increasing antibody titer, so increasing antibodies essentially in your bloodstream. Is that if you get infected initially, this takes about 4 to 7 days, 7 to 10 days for your body to kind of catch up to seeing that specific virus, bacteria, and produce antibodies against it. And then once it clears, you can see that those antibody levels dip, but they don't dip to zero. So you see that you actually have kind of this little bit that hangs around. But the little bit that hangs around is super important because these antibodies have kind of been forged in the fire of infection and are really potent and really recognize whatever is going after you. So if you get infected again several weeks later, those antibodies are already produced. Your body already knows what to do, and you can get this secondary response that's much higher than the first response and also much quicker. So instead of seeing stuff in 4 to 7 days, it can turn on 1 to 3 days, and then peak can be in 3 to 5 days. So it's very, very rapid. This is why it's also important to get vaccinated, because this is what vaccines do, is it tricks our body into thinking it's infected when it's not actually infected. So we give our body different parts of viruses in this case, and it will create this primary response, right? So you have a lag time. I don't know if you all had your COVID vaccine, if they said, you weren't fully vaccinated until like 2 weeks later, right? And that's perfectly on this timeline, is about 2 weeks for your body to ramp up its response, create memory, and then like have these very protective antibodies hanging around. And so that way, if you actually got real COVID, you would hopefully have this response where your body's already seen it, it knows to look out for it, and then it's able to ramp up very, very quickly. Yeah.
For the initial response, and IgG is for the secondary response, is they're all produced by that same B cell with a memory, but just that Y1
Oh yeah,
early and the other one late.
that's a good question. That's so this might be getting into the weeds for a lot of people, but all B cells start off producing this thing called IgM, and then once they get activated and they recognize their antigen, then they will do this thing called class switching, which they switch to making an IgG antibody. And then that IgG antibodies are really good for the things that we basically that I just described, that help cells eat things, and then also blocking invaders. There are other types of antibodies that have slightly different functions. Like there's an antibody type called IgE, which is involved in allergies, that, it does the same thing where it recognizes something, but usually it's, pollen or something innocuous, like it shouldn't recognize it, but the end bit, this kind of non-specific bit, interacts with cells in a different way. And so the IgE part interacts with these things called mast cells, and mast cells then release a bunch of histamine, and that's when you get those allergic reactions. So yeah, that's a good catch. Yes.
Thanks. Now, if it's a cold,
Yes.
when it goes back up on the right, might you start to feel sick and then everything kicks in and you actually doesn't develop, or wouldn't you feel it at all?
That's a good question. I think it probably depends, but I think you would feel a little bit sick and it might clear out. So I guess, yeah, it depends on what it is. But that's definitely a possibility. I mean, the idea is to eliminate it quickly, hopefully without getting too much, cold-like symptoms. So, yeah. Awesome. So one of the other big things about antibodies is that they can be manufactured. And in fact, they are manufactured. And that's part of what brings us here talking about cancer immunotherapy. This, for example, is a cancer immunotherapy drug on the market. The common name is Opdivo, but the drug name is nivolumab. So this is actually an antibody that's specific for these signals that cancer sends out. And next week, we'll go into a little bit more of talking about this, how this works. But these are great tools because unlike kind of small molecule drugs, and when I say small molecule, that's kind of like what we think of as a classic drug. It's this chemical that you take, kind of goes everywhere in your body, and it might bind some receptor or might bind some enzyme and block its function, and that's why it works. But even though those drugs, kind of those chemical structures are supposed to be specific for whatever they're targeting, that's not always the case. Usually there's some sort of side effect where it might bind something unexpected and then create a side effect. Whereas antibodies are so specific that they have kind of a lower threshold for side effects. So they're only really binding what they're supposed to bind and not other things. And because it's not a chemical, it's this protein, it just interacts with the body differently. So there's usually not like toxicity associated with these sorts of things. Okay. So B cells, the main job of B cells is to produce antibodies and form memory. Antibodies are the small proteins that bind to invaders with high specificity. And then antibodies can block interactions. They can activate other immune cells. They can activate proteins in the blood, all to destroy invaders. And then, of course, antibodies can be manufactured and used as very targeted drugs. So this is my realm. I'm a T cell biologist. So I love T cells and I love this. Yeah. So a T cell mounts a immune response against non-self antigens, and a Mr. T cell pities the fool who expresses a non-self antigen. So unlike B cells, T cell actually does stand for where it comes from in humans. So that's why it's a little confusing. So T cell actually, the T stands for thymus. And the thymus is this organ that actually sits kind of right above your heart. And although this is the organ that matures T cells to become kind of fully functional T cells. So T cells, like antibodies, have receptors that are very specific for parts of invaders. So that's kind of like the theme of the adaptive immune system, is that it's a very specific recognition. So again, it can recognize that like 2013 McLaren mirror, or even just the emblem, whatever part of the car it recognizes. And so this is, again, like a lock and key mechanism, except instead of where antibodies are kind of floating around, T cell receptors are locked into the T cell. So just like this, these kind of little spikes are all the little T cell receptors. So T cells can be broadly defined in these two categories. And if you've ever talked to an immunologist, you've probably heard these terms, and it's too many numbers, too many letters. It's a lot. But broadly, you can define them as CD4 positive or CD8 positive. And these just are markers that immunologists have used to distinguish these different cell types. And they do have very different functions. And so, yeah, CD8, CD4 are used to just distinguish them by immunologists. But CD8 T cells, these function as the killer T cells, are also known as the cytotoxic T cells. So it's kind of like this ninja right here. He's ready to find and kill those infected cells. So they are the soldiers of the adaptive immune system, where they can very specifically recognize infected cells or bacteria themselves and go and directly kill. Whereas CD4 T cells, they act more like generals of the immune system. And so they are more giving commands, kind of helping to set the battlefield, and don't necessarily take as direct a role in killing. So they communicate, again, through these chemical messengers called cytokines, and they kind of set the environment up so that it's favorable for these CD8 T cells to come in and kill. And by favorable, I mean these cytokines can actually interact with cells, tissue, like let's say it's the back of your throat, you have some sort of cold infection. These will tell those throat cells to start expressing these different receptors on them that make it more recognizable to these CD8 T cells so that they can more easily kill them. And so that kind of works in that way. So both are very critical and needed to coordinate successful attack on invaders. And this is actually one of the things I find most interesting about T cells, and this also goes with B cells as well, but you have millions of T cells in your body that recognize a different thing. So at any moment, your T cells, if you take out 2 T cells, they're likely not going to be the same or recognize the same thing. However, when you get infected, that T cell essentially is found, and then it explodes and multiplies into millions and millions of itself. And so you can have a lot of that. And when you have a lot of those same T cells around, you can kind of use that as a gauge of what you were infected by. Same thing with antibodies. You have millions of B cells and the antibodies that recognize different things, but it's only until that invader comes into your body that it moves and multiplies itself, and then now you have a bigger proportion of that. So how does a T cell get activated? In this example, I'm actually using an example from cancer immunology, but it's the same for being infected with a virus or any kind of invader. So as I talked about in the innate immune system, we have these cells that eat things, and they eat things not just to destroy them, but they eat things to also bring back and present to the immune system. So like I said, there is kind of this problem, right? We have millions of T cells that recognize a different thing, and we have to find the one T cell that is going to help us in the fight of whatever is invading us at the moment, right? And that's a huge task to go through millions of T cells. And so in order to do this more easily, right, there's kind of these designated meeting spots for T cells and then their innate counterparts, and that would be in lymph nodes. We've probably all heard about lymph nodes where you can feel it when it's swollen and you have a cold. That's because a lot of activity is happening in those lymph nodes. And so lymph nodes and lymph in general kind of like takes the fluid that's in your tissues and everything kind of drains into those and drains through them. And your lymph nodes will actually filter out. There's specialized immune cells that will take up things that come from your different tissues and present them to your adaptive immune system. But in this case, in the case of cancer, you might get a cancer cell that dies or is killed by some innate immune cell. And then it releases these things called antigens. And antigens are just those specific bits of invader that the immune system can recognize. So again, back to the car analogy, it could be like an antigen would be like the mirror of a car or the car emblem or the steering wheel. And so those get released, and then these specialized immune cells called dendritic cells or antigen presenting cells will actually eat those antigens, and then they'll drain into the lymph nodes. And here is where all those different T cells are hanging out, waiting to be activated or encounter their specific antigen. So this antigen presenting cell or dendritic cell will go into the lymph node, and then those T cells will kind of come alongside of it and test it. And then if they see the thing that they're specific for, they'll all of a sudden activate. And so when they activate, they divide millions and millions of times. They're actually one of the fastest dividing cells in your body. And what's interesting is that at times during kind of this expansion, they can divide even faster than cancer cells, which is mind-blowing. But that's also an important point because we probably all heard about chemotherapy, and what chemotherapy does is it targets rapidly dividing cells and just tries to kill those, but your immune system is rapidly dividing. And so that's how you can become immunocompromised when you take chemotherapy, is that it's also destroying all these other cells, and you're just hoping that it's destroying the cancer faster than it's destroying your immune system. All right. So they get activated, they divide, which is also a little bit what, why your lymph nodes swell, is because now there's millions of T cells that are dividing and then going out into the blood and then going to the tissue where the tissue that's infected, or in this case, cancer. So, right, you get in the blood, and then you get this infiltration of T cells into the tumor, and then those T cells recognize that same antigen that they were presented on those tumor cells, and then they're able to kill them. And then you kind of get this cycle, right, where antigens get released, more come back, and then you can activate more T cells, and you get this feed forward loop. So in general, T cells have three main jobs. One, they can use chemical messengers to coordinate and start the adaptive immune response. They can detect and destroy infected or cancerous cells. And then they, like B cells, can also create memory. And this can prevent reinfection, but it can also prevent potentially cancer coming back as well, which is why it's such a powerful tool to treat cancer with. So, like I said, the generals of the immune system are these CD4 T cells, and they give out their messages via these chemical coordinators. So the chemical messengers actually alert the body to what type of invasion is happening. So you can imagine that there's lots and lots of different things that try and infect us throughout our lives. And so we need kind of a customized approach to deal with each thing. So, for example, you don't have to worry too much about the names of these, but they're termed Th1, Th2, Th17, and Treg cells. But when a virus or bacteria or even cancer is detected, and the CD4 T cells get activated, they turn into what is called a Th1 cell, and then they can produce this cytokine called interferon gamma. And interferon gamma helps our body to deal with viruses, bacteria, and cancer, again, by upregulating things on cells to make it more easy to detect by the immune system and easier to kill. So to say like, hey, I'm infected with this virus, like, you should come take care of that. So this other type, Th2, it actually deals with helminths and parasites. So in the US, we really don't have a lot of like Th2 based infections because we don't get a lot of parasites, but of course you would still need something just in case you got a parasite or some like worm that infected your body. These cells are kind of specialized to take care of that. And then fungus is taken care of by these Th17 cells. So That's all good. And then these Treg cells, which I really like because that's what my PhD was on. They are natural suppressors of the immune system because all of these things that I presented so far, usually inflammation or like, and when I say inflammation, it's usually those cytokines that are signaling, right? That bring in other immune cells, and then those immune cells get activated and they send out signals, which bring in more immune cells. And you can see how kind of this is a feed forward loop. And so you need something in your body that once everything is cleared out to bring it all back down. And so that's what these Treg cells do, is that they bring everything back into balance and to maintain internal homeostasis. Yes.
I was under the understanding that the thymus gland stops growing at a certain time. Even though it stops growing, it still continues to produce T cells.
No, actually, that's a great question and good insight. So yeah, your thymus actually does stop developing at a certain
Puberty,
point in your
isn't it?
life. It's pretty early,
Yeah.
but so that means your body essentially has all the T cells it's gonna have and need for your
At that
entire
time.
life. Yeah. And so that's also kind of what's a little crazy, right? You think, why don't we just keep, producing T
Mhm.
cells so that just in case we encounter anything bad, we are ready for it. But it produces so many different things that it's specific for, including some stuff that's specific for yourself, which is how autoimmunity can arise. It kind of rides that knife edge of we want to be prepared for anything. And so we'll be like on the slight edge of over prepared, which might mean we attack ourselves at some point in our life, but hopefully everything else, takes care of that. Like Treg cells, they're supposed to take care of those self-reactive cells. So yeah, your thymus does stop growing and stop producing, and then you just have the T cells.
just
that you
have them.
Yeah.
What if that thymus gland doesn't stop growing? That can cause cancer as well.
Yeah. So yeah, you can get cancer from thymus.
Yes.
And because T cells are rapidly dividing too, there is always that kind of worry that if those get out of control, then yeah, you end up with cancer. Yeah.
Thank you.
Yeah. All right. So CD8 T cells, unlike the CD4 T cells, CD8 T cells are the direct killers, and they're the very specialized ninjas that will go and attack cancer cells or infected cells. And so when they're activated, they will traffic or move to the tissue of infection, because again, they get met in those lymph nodes, and then they have to go out and find where it's infected. And they find where it's infected, again, via those chemical messengers that those innate immune cells send out when they initially encounter whatever it is that they've encountered. So once they engage this infected cell, so again, they see the antigen that they're specific for on this infected cell, they can release these things called granzymes and perforin. So perforin sounds like perforated. It basically does that where it'll punch holes in the cell, and then that makes this channel or pore for those granzymes to go in. And those granzymes are just super reactive, and they kind of just chew up everything on the inside. And so, as you can imagine, you just gain entry and then just chew up everything, and the cell dies. So another important thing about T cells is that they also form memory to prevent reinfection. So even though they are not sending out antibodies like B cells, you do have T cells that hang around as memory. And like the B cells, they have kind of this very similar timeline that we showed before. So this doesn't have specific days on it, but essentially like this would be 7 days after infection. So let's say this activation would be you get infected with something, and then here's the cell number of these T cells. You can see it rapidly grows to control infection. And once it's controlled infection, you actually get this huge contraction. So about 95% of those cells that were produced for that infection die off, but 5% stick around as memory. And when they're memory cells, they're kind of, because they've already seen this problem before, they know the solution to this problem, they can kind of enter in and fix it quicker next time. So it's like you have some wanted posters posted around your body for specific invaders, and you can quickly recognize when that same invader comes back. And they're more powerful than these initial T cells, and they do a very good job at controlling and killing things. So, and this can apply to cancer as well. Go back here. What's cool is cool about this memory is that if this tumor cell ever gets cleared, or this tumor ever gets cleared, you may have memory cells that are formed. And so if any other cell gets mutated and starts to transform in that same sort of way, your body would then be able to recognize it and kill it off quickly. So again, this is a huge advantage to immunotherapy over conventional therapies where you're just treating with a drug, and the drug might kill off tumor, but once you stop giving the drug stops working. But the immune system always keeps working, and it's kind of biology fighting biology. Okay, so yeah.
So you just made a leap to the immunotherapy when that last comment you made. So if you look at what you described, it's kind of like a Hallmark movie version of a happy ending. It all
Yes.
takes care of it, okay? But then you brought in immunotherapy, which is adding a little drama,
Right.
you know, to the plot. Okay.
Yes.
So am I reading that correctly? That's a perfect scenario where the body takes care of itself.
Yes.
And then you introduced immunotherapy, which is here, you know, it's, you know, coming in on a white horse that's kind of save the day.
Right. Yeah. So that's a wonderful observation. And actually, I think in next slide or two, I have because if that was always the case, right, we wouldn't need immunotherapy. We also wouldn't get cancer if the immune system was perfect at recognizing and destroying, because we could do that all on our own. But we do get cancer, and immunotherapy also doesn't necessarily work for everyone. So why is that? Yeah, and I'll talk about that in a second. No, I know the Hallmark movie.
Does the body sometimes encounter challenges that T cells do not recognize? I mean, you say there are millions of different T cells, but does that cover every possible virus or bacterium?
That's a good question. Theoretically, yes, but it's hard to totally say. So what's interesting is that there are, I was reading more about this yesterday. There are essentially 10 to the 25th possible combinations of T cell and B cell receptors. So that's, I don't even know what, that's a really big number. But yeah, a bazillion. But when people actually have looked at how many actually exist in people, because that's like a theoretical number based off of all the different combinations of the genes that make these, you usually have somewhere around like 10 to 20 million different receptors in your body. And that also could be because in different societies, we kind of have common things that are encountered, that your body knows that that's going to be needed. And so like when you pass that on to your offspring, or because you grow up in actually a specific, environment, you kind of develop those, like, keep around those T cell receptors because you know you're going to encounter it. So it does kind of cheat a little bit, where it's not just like ready for anything. And I guess you could encounter something that your body hasn't encountered and doesn't know what to do with, but I feel like you'd be able, like, there's something out there, and that's kind of hard to determine, I think, too. Yeah.
So Gary asks, oh, someone else came in. Gary asks, how do evolutionary biologists explain the development of such a complex and sophisticated defense system?
Oh, man.
Their lecture.
I thought I was gonna get easy questions. I don't know, honestly. It's very interesting because not all kind of this modern immune system does not exist in like kind of smaller or like evolutionarily farther back animals. Like sharks, they still have antibodies, and then I feel like fish still have some sort of immune system. It's not quite the same as ours, but then when you start to like get kind of farther back than that, you lose a lot of the immune system. So it's a really good question, and I'm not entirely sure, but it is fascinating how it works and how it's able to recognize a lot of things.
Phylogenetically, at an earlier stage, invertebrates and so forth, they don't live very long.
They don't, yeah, and there are kind of innate defenses. It's just not necessarily immune related, like cells themselves, like different tissues will be able to fight different things in different ways. And so those are kind of like proto immune systems, if you will. And then, yeah, it developed to become more complex as animals became more complex and the needs became more complex. Yeah.
I think this kind of gets to the point that I made earlier, that we don't know a lot about the immune system. We do know a lot, as you pointed out, from what we've seen, but there's a lot we don't know.
Yes.
And, because that 10 to the 25th.
Absolutely.
Yeah, okay.
Have another question?
Yeah. This is good because we're getting towards the end of my slides, but I wanted to have a discussion, right, and have people, like, whatever you guys are interested in, I'm happy to answer.
I have a couple of questions that if I wait much longer, I'll probably forget what they were. Are we doing a disservice by propagating the antibacterial soaps, for example, that are available in stores, where we're preventing our immune system from even being, developed better, earlier type of thing? Are we doing a disservice to the immune system?
Right. I think so. In some contexts, like what I'm thinking of is there's this study between like these kids who grew up in an Amish farm versus like a really clean, like modern household, and the Amish farm kids had a lot better immune system, just because they're encountering all these different things. So I think that does have something to do with it, but also it is helpful to have those antibacterial things to help prevent infection. So it's hard to say because like our in the society that we live in, our immune systems already might be in not as developed as those in the Amish kids, right?
Yeah. And also is there anything that an average young adult or older adult can do throughout their lives to help strengthen their own immune system?
Oh, that's a really good question. So back to your point, there's a lot of stuff we don't know about the immune system. And that's actually an active area of study for a lot of people is exercise science, for example, is starting to explore into immunology and how exercise might actually enhance the immune system. And we kind of have an anecdotal sense of like, yeah, if you exercise, you live longer, you feel better, and you're more protected, but we don't know exactly how that happens. So yes, I mean, that's an active area of study. I don't really know. I'd say exercise, eat good.
And don't get old.
Don't get old. Yeah.
We do have one more quick question from online. He's asking about the difference, if you're going to discuss the difference between a primary checkpoint inhibitor such as Yervoy, as opposed to Opdivo, which you referenced earlier, a secondary checkpoint inhibitor.
Yes, and that will be more for next week. I wanted to have this week be kind of give us a similar vocabulary of what the immune system is and how things kind of work in a general sense to then understand why does immunotherapy work the way it does. So yeah, I will get to that. Yes, it does.
Is there a link between psoriasis and T cells?
Oh yeah, absolutely. Psoriasis is an autoimmune disorder. And so you have T cells that recognize parts in your skin that then, attack and create inflammation. And that's how you get the like scaly skin. So that is a kind of immune response gone awry because you are not supposed to be attacking your body. But for whatever reason, and people actually still don't know this, this is also an area of active investigation of why autoimmune disorders start in the first place. We know like how they happen in terms of what they're attacking, how the cells involved and everything, but we don't know what triggers your body to all of a sudden switch to attacking itself. So, all right, yes. So this is an answer to if the immune system can kill cancer, then why does cancer still happen? And that kind of comes to this idea of the 3 E's of immunoediting. So those 3 E's are elimination, equilibrium, and escape. So I'll go through each of these individually, and if there are still questions, we can talk about this. But essentially, in the early stages of cancer, so like I showed in that kind of one of the first slides, you have this initiation and then promotion of the cell to start dividing. And it might start dividing uncontrollably, but your immune system is actually able to recognize that something has gone wrong with this cell. It's not expressing normal proteins, and it is starting to crowd out other cells that it's not supposed to. And so this will actually elicit an immune response. And a lot of times, and actually I would say most of the time, we're in this kind of elimination phase where the immune system is actually able to detect and totally clear out any cancer that arises. However, there is also a scenario where you reach this point of equilibrium in which maybe some of these cells, as they're dividing, they acquire different mutations that make it harder for the immune system to see them or harder for the immune system to actually kill. And all of a sudden, that's like what this orange cell is that they start surviving this immune attack. And they're still able to divide because they're kind of hidden from the immune system. And so really, there's this selective pressure, kind of like the Darwinian evolution concept of natural selection, right? Where there's this huge selective pressure to eliminate or all the cancer cells that are detectable by the immune system are going to die. But if there are any cancer cells that are not detectable by the immune system, those will live and then start to grow out. And so they start to grow out, but you might reach this stage again. And this is thought to happen potentially over years. Like we might get those cancer mutations 20 years before we actually develop cancer. And so we actually might be in this equilibrium phase where we don't know what's going on. We don't necessarily have cancer yet because your immune system is controlling it as well as basically as fast as the cancer is also growing. But at this stage, it's, you know, very small, undetectable, won't do anything. But then there may be another tipping point where these cells start to maybe divide faster or are more invisible, have more defense mechanisms against the immune attack, that they start to grow out and then they escape the immune system. And so then this is when it starts to take over, and this is when you would actually get a detectable tumor. And so really cancer and the appearance of cancer, I think I have this slide, is the failure of your immune system. So your immune system, if you don't have cancer, is probably doing its job really well. That it's most of the time surveying, keeping things in check, maybe even keeping things in this equilibrium stage. But it's not until cancer cells mutate enough that they're able to escape and form these tumors. So your immune system starts out recognizing things, and then those selective pressures can push cancer to try and survive in different ways and survive that immune attack. So there are lots of different ways that cancer can put up barriers to the immune response and become kind of resistant to that immune destruction, right? So one way is that cancer cells can actually become invisible to the immune system. And by invisible, I mean the immune system and T cells specifically, they recognize this thing called MHC or major histocompatibility complexes. And these are just proteins that are expressed by all normal cells in your body. And these are kind of essentially like little windows into the cell that they hold proteins and say, immune system, look at this protein. Is it normal? Is it not normal? Can you just check it out? And so T cells and other cells will kind of look at those and say, yeah, you're good. Or maybe there's a mutated protein in that, and that indicates that it's cancer. It will, attack that. But what cancer does is it actually downregulates or turns off those MHC molecules. And so then it becomes like the immune system has no windows to look into the cell and see what's going on. And so then it essentially becomes invisible, even though, it knows that the cancer is still there. It just can't do anything about it because there's nothing being presented to it. It can't like look into the cell. The other thing cancer cells are really good at is they can produce these stop signals to the immune system. And these stop signals are kind of the essence of checkpoint blockade. We had a comment about that on the internet, and we'll talk a lot about these. Therapies have been developed against these stop signals that have been very, very successful. And so a lot of immunotherapy is kind of figuring out how and why this works. And so that's kind of depicted here, where this cancer cell is putting out these different stop signals to the T cell, and then the T cell is like, oh, okay, I'm not going to kill you. You seem like you're fine. Then cancer cells are also kind of metabolically deranged. So they take up lots of nutrients, and in turn, they produce these kind of toxic nutrients. And this is something I'm very interested in. This was my PhD work, and then some of my work now is kind of looking at how metabolites affect different immune cell function. And then they can also, so that's here, this kind of metabolic restriction. So then they can also produce signals that recruit and support these immunosuppressive cells. So like I said earlier, there's this flavor of CD4 T cells that are called Treg cells, and Treg cells are good. They're the natural suppressors of your immune system, but cancer kind of hijacks their function and then recruits them and creates this immunosuppressive environment where Tregs are kind of dominant, stopping those CD8 cells from killing the tumor. So there's also a lot of work into looking at how do we target Treg cells or these other suppressive cells to get them out of the way so that our immune system that is going to kill those cancer cells can come in and take care of it. And then also, I forget about this one a lot, but there's physical barriers that are part of like preventing the immune response against cancer. So there's a lot of abnormal blood vessels and things in cancer that prevent it from being oxygenated, but then it also prevents cells from actually getting into the tumor to do the things they need to do. So kind of back to this question, why does cancer still arise when there's an immune system? So the presence of cancer, as I said, indicates the immune response has been overcome. And then there's these selective pressures by the immune system that enriches for cancer cells that can survive the immune attack. And then cancer produces and supports an environment that promotes its growth. And so what cancer immunotherapy has really aimed at is to overcome these barriers. So next week, we're going to talk about how these different therapies address these barriers. So I kind of put it up into these categories, but you can think of, the immune system needs to be able to detect cancer, which sometimes it cannot always do. And so there are things called oncolytic viruses, dendritic cell vaccines, even radiation, and this new thing that I just found out about called histotripsy, that can all help improve detection of the tumor by the immune system. And then there are molecules that can block these stop signals, and that's generally known as checkpoint blockade, and we'll go into more about that next week. And then, of course, the metabolic environment, which I'm very interested in. I put CAR T cells in here. CAR T cells are these T cells that are kind of redirected against cancer. You can take them out of the patient's body, give them a receptor that recognizes cancer, and then reinfuse them back into the patient. So they're directed back against cancer, but they also, once they get into that tumor environment, they run into a lot of these like metabolic effects. So a lot of people are working with metabolism and these things. And then of course there's metabolic inhibitors, bicarbonate, which is used to adjust the pH of the environment, the tumor microenvironment, which actually might favor different cell types. And then of course diet, depending on what we eat, people are exploring how that might affect or improve different therapies. And then there are targeting antibodies that will target these suppressive cell types. And then there's also these kind of angiogenesis normalizing drugs that will address those physical barriers. So they're drugs that will allow the blood vessels to kind of grow normally so that you can get more oxygen and more immune cells into the tumor. So with that, I will take any questions that you guys have lingering.
I just had a couple comments. Number one is it just goes to prove that we're wonderfully made.
Yes.
And secondly, there's a whole lot going on behind the scenes that I'll never know. But I will tell you this, as a presenter, I applaud you. I'm just a lay person, but boy, this is the first time I've had anybody explain this so clearly to me.
Good.
I
Thank you.
just really thank you.
I appreciate that. I never know. This is it's a tough thing. I'm used to speaking to like, scientific audiences and so,
Yeah.
thank you.
Yeah, you really nailed it. Would it be okay if you sent out the slides to us for this particular
Yes,
lecture? Because I
absolutely.
think it might, we're not going to see you again for a week
Yeah.
and we might forget something. I don't
Yeah.
know.
Absolutely, yeah. I'll be sure to include some, yeah, there was actually another question back here. No? Okay. All right, thank you all so much.